Hello, welcome to Medical Lab Science Quick Review. And our topic is chronic myelogenous leukemia, followed by questions and answers revolving around topics in the AECP Board of Certification Study Guide. The World Health Organization classified the myeloproliferative neoplasms as those with neoplastic clonal disorders characterized by a marked increase in red blood cells, white blood cells, and or platelets. There are four major ones, of which chronic myelogenous leukemia is our current topic. These four are distinguished from acute myelogenous leukemia, characterized by more than 20% myeloblasts in the peripheral blood smear. You would not find greater than 20% blast unless you see the unfortunate patient with CML who is suffering from a blast crisis. Myeloproliferative neoplasms are characterized by panmyelosis of the bone marrow. Plus, the peripheral blood smear shows granulocytosis, erythrocytosis, and/or thrombocytosis. Usually, granulocytosis refers to the neutrophils, but also refers to basophilia and eosinophilia in myeloproliferation. Looking at a right Giemsa stain of a peripheral blood smear, something stands out. We see a broad spectrum of neutrophils of different stages of maturation, from blast to segmented neutrophil, but we also mostly see myelocytes, metas, and bands. A few promyelocytes, occasionally one or two blasts. Note the basophilia greater than 2% and eosinophilia greater than 3%. Let's start with the bone marrow aspirate and slide, taken after a clinical suspicion and initial workup. A clinical pathologist would help us in our path review by observing panmyelosis of the bone marrow, showing abnormally high cellularity, high myeloid erythroid ratio, and if we look closer, we should see many myeloblasts. We should be able to visually identify and describe the broad spectrum of neutrophilic maturation, including the myeloblasts, promyelocytes, metamyelocytes, and bands. How did this myeloproliferation happen? What is the pathological mechanism of CML? Focus on chromosomes 9 and 22. ABL1 gene codes for a protein called tyrosine kinase, which is key phosphorylating enzyme that promotes granulopoiesis in the bone marrow to produce granulocytes, eosinophils, and basophils. In genetics, a chromosomal translocation results in an unusual rearrangement and exchange of chromosomal segments. The Philadelphia chromosome is the result of a translocation between chromosomes 9 and 22, resulting in the Philadelphia chromosome abnormality in the shorter chromosome 22. It has part of a chromosome 9 attached and a BCR-ABL1 fusion gene. FYI, BCR stands for breakpoint cluster region, and ABL is the human counterpart to the viral gene Abelson-Murian leukemia. ABL gene is now known to code for the tyrosine kinase enzyme responsible for unregulated immature granulocyte proliferation. Originally in chromosome 9, now it is part of the chromosome 22 in the Philadelphia chromosome. Philadelphia chromosomes are also found in some cases of ALL and AML, so it is not exclusive or diagnostic for CML. Let's look at a case study with a 50-year-old man complaining of a 2-month history of fatigue, weakness, fever, chills, weight loss, and loss of appetite. And here's the CBC, and look at that leukocytosis, low platelet count, and the manual diff shows you this differential. What is your first impression? Our first clue is the age group, 50-year-old. In CML, we see rare blasts, few promyelos, a prominent left shift with a predominance of myelos, metas, and bands, basophilia, eosinophilia, and rare nucleate RBCs. Myeloperoxidase stain is helpful for showing enzyme peroxidase in the granules of immature and mature myelocytes, but not in lymphoid cells. So in CML, since there are neutrophils with peroxidase granules, it may show some positivity. However, myeloperoxidase stain may not be helpful in CML because we already know that it is myeloid and not lymphoid cells that are involved. Terminal deoxynucleotide transferase stain or TDT is negative. Why? TDT stain is positive in lymphoblasts, so it is negative in CML. Leukocyte alkaline phosphatase score is helpful. It's low in CML and high in leukocyte reaction. In leukocyte reaction, we could see a severe leukocytosis and left shift, raising the suspicion of a leukemia. But you don't see a lot of promyelos, myelos, eos, and basophils in leukocyte reaction. LAP score and Philadelphia chromosome easily distinguishes the two diseases. FISH or fluorescent in situ hybridization is positive for the BCR-ABL1 fusion gene. PCR polymerase chain reaction test may be positive for the translocation 922 Philadelphia chromosome. Flow cytometry, well, it's not specific because there's a broad spectrum of immature leukocytes seen with the basophilia and eosinophilia, but it may be helpful to rule out other leukemias. CML has three phases: chronic, accelerated, and the blast crisis. Another CML complication is that blast transformation to acute lymphoblastic crisis, where you're going to have TDD positive and myeloperoxidase negative. Tumor lysis syndrome is noted for hyperuricemia and other associated metabolic and urinary problems. Lysozymuria. Urinary lysozyme indicates renal tubules proteinuria, glomerular tubular damage, and renal dysfunction associated with electrolyte imbalance. Please note that lysozymuria is a complication of chronic myelomonocytic leukemia. And now for some Q&A that is similar to the ACP Board Certification Study Guide. Thank you for your time, attention, and your support in helping those afflicted with cancer.